everyone, welcome back to our YouTube channel AgriAddict. This is a lecture series for IBPS AFO mains exam and also useful for other agricultural exams. I am your mentor Thrilok Srinivas. I completed my undergraduation in Agriculture College Bapatla, Acharya Engineering Agricultural University, Andhra Pradesh. Currently I am pursuing my masters in UAS Dharwad in the Department of Genetics and Plant Breeding. I also secured an All India rank of 25 in this year's JRF Plant Science exam. Our today's topic is Mendelian genetics. As you all know, Mendelian genetics is a vast topic. So, we are dividing the Mendelian genetics into three parts and we are discussing it in each topic. In this video, we are going to be discussing about the basic terminology, laws of Mendel, chromosomal theory of inheritance, and the types of dominant relationship and lethal genes. Let's know about Gregor John Mendel. He was born in 1822 in Moravia. But one of the most important thing we have to remember about Gregor John Mendel is his paper, which is entitled Experiments in Plant Hybridization. Because firstly, no one believed his theories and his paper. But in the year 1900, when three scientists, Hugo de Vries, Karl Correns and Erich von Schermack, these three scientists independently worked and rediscovered the findings of the Mendel. After their rediscoveries, people started believing the Mendel's theory and started researching on the subject. And these are the characters that are studied by the Mendel in pea plant. As you all know these characters, I kept this slide only for information purpose so that you can remember them later. One of the main reasons for the success of the Mendel is selection of the pea as his experimental material, because pea has several contrasting characters and also it is a self-pollinated crop, so there will be no genetic error in the production of the characters in progeny, and it is large flowers, so it will be easy to emasculate and cross-pollinate. There will be less work and the size of the seed is also less, so the germination problem will also be low. And the plant occupies small space, so in a small area you can grow many subjects as an experimental material. Some of the important terms you have to learn before entering into Mendelian genetics. Character and trait. Character means distinguishing feature of an organism, whereas trait means distinct variant of an organism's character. To know the difference, let's see. Take two plants, one tall and the other is short. In genetics, if we have to describe the characters of the plant, we will list them as height of the plant, girth of the stem, etc. But if we want to express traits of the plants, we will elaborate the characters as what is the height of the plant, like they are 3 meters or 2 meters, or they are tall or short. Like this, the variation in the character we call them as traits in genetics, whereas the character means the features of the plant which are present. So I think you all understood. The next point is gene. Gene is the fundamental unit of the heredity, which is passed from the parents to their offspring. Remember this point, the fundamental unit of the heredity, because it might be coming as a bit. And the other thing is alleles. Alleles means, let us take a chromosome. You all know chromosome, right? In this chromosome, this we call it as genomic position, because in this position we are having one gene, and this gene contains a pair of alleles. They might be same or different. So alleles means one or two more versions of the DNA version, means one or two more, like they might be same or different, which are present at a given genomic location. In a genomic location, there will be a pair of DNA sequences which may be same or different. We call those pair as alleles. And the next is dominance and recessive. So these are the pair of alleles I have, and the character that is exhibited is tall, but capital A should exhibit tall, small a should exhibit dwarf. In the progeny, the character that is expressed is only tall. The dwarf character is not expressed by the small a. So that means the allele which is expressed and masked the other allele's character, we call it as dominant. Means one allele which is masking the effect of the other, we call it as dominant. Here capital A is masking the small a, so we call the capital A as dominant, whereas small a we call it as recessive because it is not expressed when both the alleles are present, and the recessive will be expressed only when both the pair of the alleles are recessive. Means in a progeny both the alleles should be small a and small a, then it will be expressed dwarf character. This type of condition we call it as recessive. Homozygous and heterozygous. We all know that in a gene there will be a pair of alleles. They might be same or different. Homozygous means having same pair of alleles, whereas heterozygous means having different alleles in a pair. And homogeneous and heterogeneous. Homogeneous means in a population, let's assume there are 3 individuals. First individual has capital A and capital A. Second one also has capital A and capital A, and the third also same. Means the pair of alleles that are present in each person is not differing from individual to individual in a population. This condition we call it as homogeneous. You can see a population where they have alleles for same genes. But heterogeneous, what is heterogeneous means in a population if they have different persons with different alleles like capital A, capital A, capital A, small a, small a, small a. Here you can see the alleles are differing from person to person. This type of population we call it as heterogeneous. And phenotype and genotype. A gene has 2 alleles which is expressing a character. The character that is expressed by the gene we call it as phenotype, whereas the arrangement of the alleles in a gene we call it as genotype. You can see the observable traits or characteristics of an organism, means the character which is expressed in an organism we call it as phenotype. The genetic constitution means the arrangement, whether the alleles are different or same. This we call it as genotype. The genetic constitution of an organism we call it as genotype. Monohybrid cross. First learn what is hybrid. Hybrid means we have 2 parents which gives us a progeny which we call it as F1, means first filial progeny. Here this F1 has characters from or alleles from both A and B. So we call this F1 as hybrid. Hybrid means the progeny which is acquiring alleles from both parents. We call it as hybrid. Monohybrid means A and B, let's assume they have 5 characters. A and B has 5 characters. Both of them exhibit same expression in 4 characters. Means if B is 1 meter height, A is also 1 meter. If the color of the flower is pink, in B also same. But they will be differing only in 1 character. 4 characters are same, 1 character is different. And now let's assume This one character is governed by capital A in A and small a in B. This we assume as shape of the seed. In the F1, the genotype will be capital A and small a. Means the hybrid is having both the alleles. Means they have capital A and small a from the parents. So it will be a hybrid in the character of shape of the seed. A cross between two organisms which is having variations of a single genetic trait. Means here it is having the both alleles from A and B in the character of shape of the seed. This we call it as monohybrid cross. Dihybrid cross means nothing. Here we thought that only one character is different. If two characters are different, like small a, small b, capital A, capital B, then the F1 will be having alleles from both the characters, capital A and B, small a and B. This we call it as dihybrid, because hybrid is having alleles of both characters from the parents. This is dihybrid because parents are differing in two characters progeny is having contrasting alleles from both parents. This type of cross we call it as dihybrid. If there is trihybrid, it means another character is also present, like capital C, small c, like that. Based on number of characters, we can multiply the cross, like trihybrid, polyhybrid, etc. Test cross. Test cross is nothing but to test the zygosity of the progeny. Means we have a tall plant which is expressed by allele A, but in a organism we are having a pair of alleles. Let's assume one is A and the other one is X. We don't know whether it is capital A or small a. How we can know whether the progeny we have is capital A or capital A small a is using the test cross. Means we will take a recessive parent which is having small a and small a. Then we will cross this progeny and this parent, like small a. Based on the result, we will determine whether the progeny is capital A or capital A small a. Means the cross which is done to test the zygosity of the progeny, or to know the zygosity of the progeny, we call it as test cross. Back cross. Back cross means cross between progeny and one of its parents. Let's suppose this is a cross. One parent has capital A, another one has small a, and the progeny is capital A small a. Crossing this progeny with either capital A or small a, we call it as back cross. And the next one is reciprocal cross. Reciprocal cross means, let us assume we will do two crosses. First cross, second cross. In the first cross, we will take female as A and male as B. Whereas in the second cross, we will take the female as B and male as A. Now, the second cross will be the reciprocal of the first cross, whereas the first cross will be the reciprocal of the second cross. Means reciprocal means nothing. It's just the inversion of the genders from cross to cross. In the first cross, we took capital A as female, whereas in the second cross, we inversed it into male. That's it. you can see monohybrid cross means having different variations of a single genetic trait. Dihybrid means two genetic traits. Test cross to determine whether or not the F1 is heterozygous or homozygous. And the back cross, cross between F1 and one of its parents. And reciprocal cross, which means using them in turn as female parent and male parent, like B into A or A into B. Now the laws of the Mendel. First, let's know the law of dominance. Law of dominance means we all know that a gene have a pair of alleles. Here, the allele which is masking the effect of the other allele is called dominant. That is the law of dominance. Here you can see when two contrasting alleles are present in an individual, dominant allele expresses itself and masks the recessive allele. Means here two contrasting alleles are present as a pair, capital A and small a. They are contrasting. Now, if the capital A is dominant, it should express only its character as tall and it should mask the expression of the small a. This we call it as law of dominance. And next one is law of segregation. Law of segregation means same example. Capital A small a. In a gene we have capital A small a. Let's take other progeny. It is also having capital A small a. While the production of gametes, the parent will produce capital A and small a in equal proportions, and the production will not be dependent on one another. Means the production of capital A gametes is independent of the production of small a gametes. This we call it as law of segregation. Here you can see each character is controlled by pair of alleles present in the gene, which separate during formation of the gametes. Here you can see the alleles are separating during the formation of the gametes. And the next one is independent assortment. Independent assortment means inheritance of one trait will be independent of the inheritance of another trait. And there will be a condition. That condition is these two traits should be located on different chromosomes. Means this is one chromosome, this is one chromosome, this is a parent. Here you can see capital A small a, here you can have capital B small b. Well, the production of gametes, it will be producing four types of gametes in equal proportion. It is capital A small b, capital A small b, small a capital B, small a small b. These should have been produced in equal proportions if A gene and B gene are present in different chromosomes. That is law of independent assortment. Means capital A and capital B will be segregated independently. The production of capital B is not dependent on either capital A or small a. It will be segregated independently because they are present on the different chromosomes. If they are present on the same chromosome, the condition will be different, which we will learn in future slides. These are the exceptions to Mendel laws. These we will discuss in the further slides and further parts. They are incomplete dominance, codominance, epistasis, multiple alleles, pleiotropy, lethal genes, linkage, polygenic inheritance, and environmental factors. All these things we will be discussing in the further slides. Chromosomal theory of inheritance. Chromosomal theory of inheritance is developed by Walter Sutton and Theodore Bowery. This theory is nothing but they are stating that genes are present in chromosomes, and the chromosome is the genetic material or the DNA which is passed from parent to offspring. These 2 points are the main things of the chromosomal theory of the inheritance. Here you can see chromosome is the carriers of the genetic material. They are responsible for Mendelian inheritance. Means this chromosome is having capital A, small a, this chromosome is having capital B, small b, then this will follow Mendelian inheritance, means these will follow law of segregation, law of independent assortment, as you can see in this diagram. Here you can see they are having capital A and small a, and they are segregating in the meiosis 1 and meiosis 2 as per the laws of the Mendel. And one of the main claims of this theory is chromosome is the genetic material. I already told you chromosome explains Mendel's laws, and the chromosomes comes in pairs. As you all know, we are having diploid organisms, which means chromosome come in pair, and chromosomes vary, means if humans have 23 pair of chromosomes in a haploid set, means first chromosome will be different from second chromosome. Like that, each 23 chromosomes will different in size and shape. This is the type of dominant relationships. Complete dominant means, as you all know, capital A and small a. Here, if there are capital A is tall character, small a is dwarf character, in the progeny only tall is expressed, dwarf is not expressed. That is called as complete dominance. Whereas incomplete dominance, they will come to a compromise. Like, suppose we will go to a shop with our mother. The shopkeeper says the cap is 100 rupees. Let's assume you're purchasing cap and the cap's cost is 100 rupees. Your mother bargained it to 50 rupees. After hours of bargaining, the cost will be finalized to 75 rupees. This we call it as incomplete dominance. Means either shopkeeper's rate or mother's rate is finalized. There is a compromise or the incomplete dominance. This we call it as incomplete dominance. I am just using general example. Here the example they given is that there is a red flower is a dominant trait, white flower is a recessive trait. But in the heterozygous condition, the trait that is produced is pink in color, means neither red is produced nor white is produced. Pink color is produced. Whereas codominance means in the blood groups you all know. Codominance means as a pair of alleles there have to be two. In the blood group we have IA, IB and IO. If in a progeny IO is present, we have O blood group. IO or IA is present, we have A. But if we have A and B, then we have AB blood group. And in the B also B and O or B and B, then it will be B only. But if A and B both are present, then it will be AB. This means here both the phenotypes are expressed. If A and B are present in a progeny, A's character is expressed, B's character is expressed. Both of them will be expressed. Then we call it as codominance. And multiple alleles means, till now we learned that there will be two contrasting alleles for a pair. Like either it can be tall or it can be dwarf. But in the multiple alleles, we have more than two types of alleles. For example, let's take only blood groups only. Here we have O, A and B. But according to the complete dominance or the laws of the Mendel, we should have only A and B or A and O, because a character is governed by only two contrasting traits. But in the multiple alleles, we will be having more than two. Here we are having A blood group as one character, B blood group as one character, O blood group as one character. Means it is governed by three alleles. So like that, this multiple allele is also an exception to Mendelian genetics. And the other thing is pleiotropy. Pleiotropy, for the example, is sickle cell disease, because this one character is governing many phenotypic characters. Example, you can also take our general example as the mood of our mother. If our mother's mood is good, she will never disturb in our sleep. She wakes us up and gives us a nice cup of coffee or tea and she will give us good tiffin. If our mother's mood is bad, then it's very bad. We will be woke up from our sleep, no good tiffin, etc. Like that, in the sickle cell disease also, if the sickle cell disease is present in the person, he will be facing many symptoms, which means a single character is affecting multiple phenotypic characters or a single gene, because this character is produced by a single gene. The single gene is affecting multiple phenotypic characters. This we call it as pleiotropy. And the types of lethal genes. Lethal genes means if a person is having a lethal gene, then he will be dead. That's what we call it as lethal genes. Means the genes which are lethal or dangerous to the organism. Lethal genes means 100% the person will be dead. No alive or no person will be alive. Semi-lethal genes means only greater than 90%. Means in the 100 people there is a chance that 10 people might survive, or less than 10 people might be surviving while having these genes. For suppose in the lethal genes, if we have a gene A, 100 people will be dead due to the gene A. And in the semi-lethal genes, the B, if they have the B gene in 100 people, more than 90 people they will be dead. But there is a probability that 10% will be survive. Means it is not completely lethal. Means semi-lethal, not completely lethal. And this is subvital genes. Subvital genes means less than 90% will be dead. Means the chances are increased. So they categorize it as subvital genes. And the vital genes means 0% effect on the survival of the person. Means let's assume this is a C gene. It has no role in the survival of the person. Either if he is dead or alive, it is not due to this gene. And the other one is super vital genes. These are increasing the survival of the genes. Means you can see lethal means cause the death of an organism. Semi-lethal means death of more than 90% of the individuals. Subvital means less than 90% of the individuals it cause the death. Whereas vital genes, no effect. Super vital genes increase the chance of the survival. For example, disease resistance genes. If in a plant there is a disease resistance, suddenly the disease is affected in the entire field, then everything will be dead but except this one, because it has the disease resistance gene. So this will be increasing the chances of the survival. So we call it as super vital genes. And these are some of the other lethal genes. We call it as gametic lethal genes, which means the gametes produced like A or B. These are the gametes produced by the parent P. These will be dead because of the gametic lethal genes. If the parent is having gametic lethal genes, then the gametes it produced will be inviable or dead. And recessive lethal genes means, let's assume small a is the lethal gene, and the lethality means the dangerousness of the gene will be seen only when both the pair of the alleles are small a. If it is present, the lethal gene is present in an organism in small a, only then the person will be, or the organism will be dead. Dominant lethal genes. Dominant lethal genes is the reverse of the lethal gene. Means either the one allele or both the alleles. It doesn't matter. Capital A or capital A small a. the organism will be dead. And conditional lethal genes. This is somewhat tricky. Conditional lethal genes means either the gene is dominant or recessive. It doesn't matter. It, it will be expressed only in a specific condition. Suppose let's take A as the lethal gene. Capital A is present in the organism. Let us suppose the condition is 100 degrees Celsius. When the temperature of the environment the organism is present reaches 100 degrees Celsius, then only the lethality of the A gene will be expressed. If the temperature is less than 100 degrees Celsius, not expressed. That's we call it as conditional lethal genes. Here you can see gametic lethal genes means inviability of the gametes. Recessive lethal genes means expressed only in homozygous state. Dominant means in both heterozygous and homozygous state. And the conditional means express only in specific environment or condition. Okay, so that's all for this part. In the next part, we are going to discuss about the gene interaction, types of gene interaction, qualitative and quantitative characters, multiple factor hypothesis, expressivity and penetrance, cytoplasmic inheritance, and maternal effect. This part is going to be a long video, but I will try to cut it short. And this is our new program Vijeta, which is useful for the IBPS AFO mains exam 2024. In this program, we are having 50 plus standard mock tests, previous year papers and solution, and one-to-one doubt clearing session. And this PPT will also be available in this Vijayata program. And this is prepared by the top rankers of the ICR PG exam. So please subscribe to the program and achieve your rank in IBPS AFO mains exam 2024. So that's all for today. Thank you. If you have any doubts regarding Mendelian genetics or today's video, please post your doubts in the comment section. We will solve them as soon as possible. See you in the next video. Bye.